Hello, I'm Ashton. I'm a senior undergraduate student working under Dr. Joseph Bonello. In the Bonello lab, a main focus of our group is looking at tuberculosis. Tuberculosis is a bacterial infection caused by Mycobacterium tuberculosis. Tuberculosis is a highly contagious disease. Hello, my name is Ashton. I'm a senior undergraduate student working under Dr. Bonello. The main focus of the Bonello lab is tuberculosis. Tuberculosis is a bacterial infection caused by Mycobacterium tuberculosis. TB is highly contagious. Approximately a quarter of the global population estimated to have been infected with TB. This is a challenge to deal with, mostly due to the complex cell wall structure. The cell envelope, probably a better word, of MTB contains four main layers. This includes the inner layer, the AGP, which is the peptidoglycan galactan complex, the outer membrane, or commonly referred to as the micromembrane, and the external capsule. For the scope of this project, we're mostly concerned with the micromembrane and the interactions that occur within it. One such area of interest is the MMPL3, which is a flippase enzyme that transports necessary precursor molecules across the inner membrane into the micromembrane. And these precursor molecules include trehalose mono and trehalose dimethylate. And these are long chain fatty acids which are esterified to disaccharide trehalose. And it's pictured the bottom left here. If the highlighted R group is just another OH group. And so this pathway is a target for us because if we can fully understand how MTB builds its resilient cell wall, we can then begin to develop better or even improve existing therapies in treating TB. In order to do this, in order to map out the pathway, we need to modify a known substrate of MMPL3, which is trehalose. Now, again, pictured on the bottom left is our three click handles that we're going to selectively add onto the 6' position of trehalose that we will later be used to add a fluorescent tag. And after incorporation into stigmatus or M-marinum, we can then gather information about how these analogs behave within the pathway. Now, four different synthetic routes were explored in order to selectively modify the 6' position. And this was deemed to be a challenge due to the symmetry of trehalose as well as its heat sensitivity. And now while all four of these routes here were successful, just due to time, I only want to highlight a few things about route A, C, and D. Firstly, A is our protected SN2. And after fully protecting trehalose, we exploit the 6 and 6' increased reactivity. And we know that these positions will be the first to be deprotected since they are the primary alcohols. And so what we can do is we can limit the amount of the tolyl chloride in the reaction runtime here in step 2. And this will ensure a single addition product, although we still see a very slight amount of double addition. C is our improved unprotected productive elimination. While A worked for both the hydroxyl and the methyl hydroxylamine, which is our R1 and R2, C was found only efficient for the methyl hydroxylamine, which again is R2. So C is only applicable to our N-methyl hydroxylamine. And now C highlights a much more optimized two-step reaction with a higher yield than A and no need for a protecting group again. Now D is our modified Appel reaction using NBS instead of CBR4. Here again, selectively modifying the 6' position by limiting both reagent and runtime. And this route represents mastering of the selectively modifying tetrahalo 6' position with a 81% overall yield. This 81% overall yield is compared to C's overall yield of 17%, A's overall yield of 6%. Although we did not discuss it, just for complete context, B has an overall yield of only 3%. And now each of these routes showcase the synthetic challenges of developing each of these three probes and the methods that were employed to overcome them. And in doing so, we saw improved ability to selectively modify 6' position in the form of improved overall yields. We also were able to achieve a protecting group free synthesis for the N-methyl hydroxylamine halos. And equally as important, all three of these analogs were successful in metabolic incorporation, which is imaged in the center bottom. And I want to acknowledge the funding that was able to make this research possible, as well as give thanks to everyone in the Vanilla group. Thank you.